And now I'd like to introduce you to your hosts, Sarah and Colleen.
Hi, everybody.
Hello.
I'm Sarah Talarovic. I'm a biology staff scientist here at Vernier. I'm a former college professor. I taught for almost 15 years, different levels of biology. And Colleen McDaniel is joining me. She is an educational technology specialist for both biology and environmental science. And so she's going to be my trusty co-pilot for our training today. So the first thing that we'll do actually is Colleen is going to administer a poll because we'd actually like to know a little bit more about who is joining us in our audience today. So Colleen, would you go ahead and administer the audience poll?
Launched.
All
So there are
right.
just two questions there, just to get an idea of what level everyone teaches out there, and kind of an idea of exactly what you're currently experiencing instruction-wise. Just so it gives us kind of an idea of what state we're all in and how everybody's doing with it. All right, got a few more participants that have come in here, so just gonna let it go for a little bit longer. All right, I'm gonna go ahead and end the poll now. So it looks like we have about 80% of you teaching high school, and of that, just over 50% are doing a hybrid mix, 20% doing 100% in person, and 20% doing 100% remote.
All right. All right, thank you, Colleen. So what we're going to start with, we're going to be walking through a lab, a kind of a classic photosynthesis prep that we have that's called Photosynthesis and Respiration. It's from the book Biology with Vernier, and it's lab number 31. So some of you may be familiar with it. You're certainly welcome to follow along at home if you have equipment and if you have graphical analysis, but you don't have to. We're going to demonstrate it, and we'll start at the beginning. So I'm going to change camera views here. And the first thing that we're going to do is going to be to wrap our chamber. We have a BC 2000, which is our large biochamber, and we're going to wrap it in foil. I prefer the light gauge foil because it's a little easier to manipulate. We're going to separately wrap the lid. We don't want to wrap it all the way around the lid because we want to be able to put it on, but we're also going to poke 2 holes where the grommets are. So we're going to make space to add those sensors. All right, our next step is going to be to add some spinach. And what I have is spinach purchased yesterday at the grocery store, and it was kept cold overnight. We've removed some of the stems that are on the leaves, and we're just going to make a single layer of spinach leaves on the bottom of this container. So the spinach is still nice and crisp. I'm just going to arrange the leaves. If any are wet, you want to blot them dry, just to help cut down on any moisture in the chamber. All right, that looks pretty good. We don't want to have a whole lot of overlap. All right, so we've got just a single layer of spinach leaves. Now we're going to put the lid onto the chamber. And then we'll arrange the wrapping so that there's no light getting into that container once the sensors are added. All right, our next step in our lab is to launch the graphical analysis app and connect the sensors. So we'll do that next. All right, hopefully you're seeing my screen with graphical analysis. We're going to select sensor data collection because we're just going to do the basic live data collection for the lab. And we're going to press the power button on each of these sensors. I have a CO2 sensor and an oxygen gas sensor. And notice that they both appeared in my menu. So we can connect them. The order doesn't matter very much. All right, and now I'm going to just place them into the grommets in my chamber. And then we're going to finish setting up for data collection here. So for this lab, we're asked to change our time mode. So we're going to go down to that bottom corner, click on mode, and we're going to change our time units to minutes. We're going to change our data collection rate to 15 samples per minute. And then what we want to do is, the lab actually has you set 15 minutes. We are going to set it for 10 just because we've got a short time together today. We want to make sure that we leave some time to talk about other things. So we should be able to get away with a 10-minute data collection run. All right, so our graphical analysis is set up for data collection. What we're going to do now is, I'm going to change our view back. I'll stop sharing my screen for a little moment here. And we're going to give this a little bit of time to wait, but I'm going to set it up over here with our other camera. Now notice that we have a light ready to go for the second half of this experiment. We also have a heat sink ready to go. I'll talk a little bit more about those in a moment. But for right now, we're going to give that about 5 minutes to equilibrate. In the meantime, I'm going to take you back over to our data collection screen. And I was going to show you a few different features of graphical analysis. So while we're waiting, I can show you different views. This icon up here for view options, it looks like a stack of boxes. If you click or tap on that, you're going to see that you have the option to view different graphs. You can also look just at a data table or the graph with the data table. So you can toggle on each of these settings as you like. You also have the option to view 1 graph, 2 graphs, or 3 graphs. We're going to leave it on 2. You can also see the meters. So if you'd like to actually see the live readout of your meters, you can do that as well. We'll go ahead and turn those 2 off for right now. Notice that these buttons at the bottom are also meter buttons. So those can also show you your live readout. If you look at the bottom of the screen, they're just a little bit smaller. I'm going to also change our units. Since this lab has us change the units on both sensors to parts per 1,000. And so now we're set to collect data when our 5-minute period is ended. Colleen, did we happen to have any questions yet?
Yes, actually. So you said you're using spinach leaves. Can you use other types of leaves, or do you recommend just doing spinach?
We definitely recommend spinach simply because it's very easy to get a hold of, and it's also pretty reliable. We typically get the tubs of spinach from the grocery store that aren't treated with any type of preservative, so the ones that are fairly crisp. You can also use, I have done it with lettuce. I had actually a customer call and ask about using one of the live heads of butter lettuce, and reported back to me later that day that it worked really nicely. And I have also done it with a leaf or two of lettuce, as long as you get really fresh lettuce. I've tried kale as well. The big thing is you just want really fresh material. I have had good luck with maple leaves as well, because they're large and easy to fit into the chamber. But we usually consistently use spinach simply because it's easily accessible, and we know it's going to be a pretty reliable source of plant material.
Thanks.
Anything else?
Another question is kind of on the same note. Could you do this with an entire plant? So you said you used a head of butter lettuce, or a customer had. What about a whole like potted plant, like a jade plant or something?
So one of the problems with using an entire plant is the soil. And of course, soil microbes undergo cellular respiration. So if you try to capture this in an entire plant, you might find that the soil respiration rate is actually higher than what you're able to detect with the sensors for photosynthesis. So oftentimes you get what looks like a flat line, even though there's obviously lots of activity going on in your chamber. You could maybe try something like an air plant. That might be possible. I haven't tested it specifically, but you do want to be aware that other plant tissues, such as stems and leaves, are always going through cellular respiration and not necessarily undergoing any type of photosynthesis. So sticking with just leaves definitely guarantees, or at least increases your chances of getting a really nice run of data showing photosynthesis. We're gonna actually first do a run of cellular respiration in the dark. Yeah, doing a whole plant is a little bit problematic because of, depending on the amount of soil that you have, you may not see particularly strong results. But thank you, that was a great question.
That's all we have for right now.
All right, no worries. We are actually almost done with our wait time. All right, so it's time now for us to collect some data. All right, so we're first doing our dark run, which should be giving us cellular respiration. And you can see that our scales are vastly different for our concentrations of CO2 gas in the atmosphere versus oxygen gas in the atmosphere. Oxygen is in a much higher proportion to CO2. So it's good to have them on two separate graphs, even though we could technically put them on one. But if we do that, we're not going to see a real, we're not gonna be able to visualize the data as nicely, just because the scales are so different. And if you notice, as we start data collection, it looks a little bit boring because we were very zoomed out on the view of our data. So if we use our zoom button here, we can actually zoom in on the data as it's being collected, which gives us a little bit more dynamic of a view. So you can see that our CO2 levels are already increasing. All right, well, we're going to have about 10 minutes as we watch data collection. So what I thought I might do is show you where to find some things on the website. So I wanted to introduce you, if you're not familiar with our remote solutions, we have a number of different solutions available for teachers. And you can find it right here on the main web page. There's a box at the top that says Explore Remote Solutions. So if you click on that, that's going to take you to different levels of education. So let's go, I think our majority of our audience was high school. So we can scroll down through some of these different options. Note that we have a number of webinars already online. And one of the things that we'll demonstrate today, we're currently using graphical analysis, and we have an upgraded version called Graphical Analysis Pro. Colleen and I will be demonstrating some of the features of Graphical Analysis Pro a little bit later, but you are welcome to sign up for a free trial. We have some other options. Note that there are things like pivot interactives. We're not going to demo any of those today, but I wanted everybody to be aware of the different options that are available to teachers. We also have some free options, including our sample data library. So you can use Graphical Analysis or Logger Pro, and if you need data to share with your students, if you're unable to collect data, you can pull a number of labs from our sample data library. So this is a browsable index of labs. And the first page that opens is biology. And notice that we have them listed by our books. So if you're browsing for some material in a particular book, not everything is here, but quite a few options are here. And what they are data files. In some cases, we also have a video that goes along with it, just to introduce the concept and introduce the type of sensor being used. But notice that we have a lot of labs listed. Try to scroll down here, there we go. So we have quite a bit from biology with Vernier. Those are using graphical analysis since that's our newer software. We also have some labs from Human Physiology Experiments, Volume 1, as well as Volume 2. We also have some from older books using Logger Pro. But we have quite a few options. We also have sections for chemistry, for physics, and then also for elementary science, middle school science, and physical science. So those are data files. You can link to both the lab instructions written for the student, as well as the data file. And that way, if you don't have a way to get live data to your students, you can send them a data file and still have them analyze it. So again, you get there just by going to our main webpage and then clicking on remote resources and following the links to the data library. All right, so we'll return to our data collection here. Our oxygen is not looking particularly dynamic, but our CO2 is looking pretty good. And this does sometimes happen with oxygen.
Sarah, why would that happen with the oxygen?
There's a number of different factors. The oxygen sensor is a little bit more sensitive to temperature changes. It's also a bit sensitive to humidity. So if the spinach is a little bit moist in the chamber, sometimes you don't get a particularly dynamic shift. Also, the oxygen sensor, I forgot to mention that we highly recommend storing it at all times and operating it in an upright position. I actually have been holding them in a beaker. Let's see if I can change my view here. When I'm getting ready to use them, here's our overhead view. I actually have them sitting in a dry beaker. And the reason for that is that there's an electrochemical gel inside the sensor that uses oxygen and it breaks down over time. So when you first receive your sensors, they're in a box with a big arrow printed on them. And that is to remind you to store them upright. So you want and with this Go Direct sensor, you want that USB port pointing up. And that's because that electrochemical gel will shift. And in doing so, it can throw off your results. And note on our CO2 graph, we have a little jog in our data. That's actually our temperature correction that is kicking in as our temperature cools off a little bit in the building. We're still a little bit adjusting to the cold nights here. So it was a little bit warm in the room when I came in. But the CO2 sensor actually has live data correction for temperature changes. And that's all that is, that little shift up there at about minute 6.3. That's going to be just one of those small temperature corrections. Some teachers do prefer to do this lab with just the CO2 sensor, which is perfectly fine. The O2 sensor does have a limited lifespan. It typically, we expect it to last about 6 years if it's stored properly. They can be refurbished. So we are able to put a new electrochemical cell in sensors for you. So if you have some older oxygen gas sensors, please don't hesitate to contact us, see if they can be refurbished.
Sarah, another question.
Yeah.
What about using a different container? Could that affect the results or improve results possibly?
Oh, absolutely. So we can do this lab actually using a smaller container, such as the BC250. That is available for you. And the oxygen sensor fits very nicely in the gasket. And the CO2 sensor can fit in the neck of the bottle. That gives you a small chamber, in which case you don't want to crowd the leaves. You want to gently place them in the chamber. The other option is to make your own container. And with these Go Direct sensors, if you're using them on Bluetooth, you can actually use a container that completely seals. Again, you want to keep your O2 gas sensor in the upright position at all times. So you want it to be vertical. All right, it looks like data collection is finished. So let's quickly change that out. I'll show you some data analysis, but we'll get our 5-minute wait period set up before we come back and do some analysis. So give me just a moment, and we're going to remove the aluminum foil. We don't want to open the chamber. We just want to take the foil off. So in relation to your question, Colleen, about the chamber, we do actually sell these little grommets, these rubber grommets that fit the sensors separately. If anybody wants to create their own chamber and cut holes, you can absolutely do that. All right, so we're gonna now return to our setup over here, but we're gonna turn on a light and we'll talk a little bit more about how the heat sink works. Let me plug in my light source. It's going to be a little bit of an abrupt change. All right. So that is, I just realized our camera angle got a little bit shifted.
Sarah, while you're doing that, we actually had a question come in.
Oh, fantastic.
Can students download their own copy of GA?
Oh, yes, absolutely. So students on our website can get, let me take you back over to our website here. So if you send your students to our website, you can either give them the direct link to our downloads page, or you can just tell them to go to downloads. But right here under downloads is going to be the first choice is going to be graphical analysis. And that is available for free on 5 different platforms. So depending on what types of devices your students are using, it's pretty unlikely that they wouldn't be able to run it, because we have it for Windows, we have it for Mac computers, we have it for Chromebooks, in which case they would get the Chrome Web Store version. We also have it available in the App Store for iPads or even iPhones. And then we also have it available for Android devices, tablets or phones. And that would be from the Google Play Store. So if your students need it, it is available for free, and it's very easy to download and install. All right, I've started our 5-minute wait period, so we're just gonna wait on. Any other questions about graphical analysis, since we have to wait a few minutes for our chamber to equilibrate?
And just so we clarify, this version of graphical analysis is free. So that's why students are free to download it as they will. The big thing is if the students are using a school issued computer, Chromebook, or other device, you might have to get your technology department involved, because a lot of times they have different, they don't have the rights to actually download something onto their computers, especially if it is a school issued one.
Yes, that is correct. We do have there is an MSI installer. If your IT department asks you for the installer, there is a way to get that. If in doubt, you can always contact us at either support@vernier.com or biology@vernier.com, or you can give us a call, or you can use our chat feature, because we also have a chat feature on the website. And our number is 1-888-VERNIER. So I'm going to put our meters up just so I can keep an eye on them. So with that light on, that's an LED grow light. And I mentioned that I would talk a little bit about that heat sink. So that's what we recommend in the book, is to use a beaker, a 600 ml beaker filled with water, and that's totally fine. Just for space issues today, because we had kind of a small area for the camera, I used a tissue culture flask, and you may find these laying around if you have a lab at your school, especially a university lab. You might find these big flat chambers. Those are just plastic tissue culture flasks, and they work really nicely as a heat sink. The LED light does not get particularly hot, but other lights, if you use an incandescent bulb or if you use a halogen, they tend to heat up quite a bit, and the heat can actually disturb your prep. So I like using the LED bulb simply because of the lack of excess heat. It does produce a tiny bit, but not very much.
So other light sources that could be used are halogens and incandescents. What about compact fluorescent lights?
Oh, compact fluorescent lights. We've actually studied those a bit in the lab, and we have found that the compact fluorescents just don't put out enough wavelengths in the red and blue range for plants. So you don't get very good results with compact fluorescents. So if you're familiar with those are usually kind of a twisty looking bulb. Sometimes they'll put a shell over that to hide the little twists in the tube, but those just don't produce particularly good results. And when we've analyzed them in different ways, we actually did an experiment where we looked at the light output, and we found that the wavelengths being output are mostly in the yellow and green range, which just isn't terribly useful to plants. All right, so it looks like we're ready to start data collection. My 5-minute alarm just went off. So I'm going to go ahead and start data collection for now we're doing our light run for photosynthesis. Oops. There we go. Wanted to get back to just my graph screen there. So you can see our CO2 level is already dropping pretty drastically. Yeah, the LED lights are getting much more easy to get a hold of. The prices are pretty good. They cost about the same, depending on where you find them and what quality you get. The cost is not much more than purchasing compact fluorescents. So they do make very nice, and they do last a long time as well. So they do make nice additions if you have the inclination to get a light. It's also, I'm going to demonstrate over here on my prep that I can touch this light and not burn myself. And that is a nice feature when you're working with students. That's a fairly bright LED grow light. You can find smaller ones that are a little bit cheaper. But that light you could probably share between several preps even. You can back it out a little bit and still get very intense light. Any more questions, Colleen?
One is, can you do this aquatically? Like, is there a way to do this experiment with aquatic plants?
Yes, absolutely. You would want to use a different sensor. So rather than using the gas sensors, and actually if we use this other screen, you'll notice that on our gas sensors, we actually have a little icon that indicates that it is not to go into water. That is one of the most surefire way to ruin one of these gas sensors is to dip it into any kind of water. So we don't usually recommend that you use these over an aquatic environment at all, and certainly don't dip them in. But we do have a sensor that is made for that, and that is the GoDirect optical dissolved oxygen sensor. So this sensor can be used in an aquatic environment. It is also water resistant, so it can be splashed. The sensing tip is down on the bottom. This is just a storage bottle that has a moist sponge in it. And then it has a sensing tip on this end. It also has, this metal ring is the temperature gauge. So it's basically the thermometer is built in here. And it's also pressure compensated. So if you need to do anything in an aquatic environment, this is going to be the sensor that you want. We have done this prep in a 250 ml Nalgene bottle using aquatic plants such as Java moss and Christmas moss, which are, those are both aquatic mosses that are available for home aquariums. They're very popular with fish hobbyists. And it makes a great prep to do if you use the dissolved oxygen sensor. Thank you. That was a great question, Colleen.
We also have someone that's getting ahead of us here who wants to know what the video does in VA.
Okay. We'll definitely show that off in a moment. While we're waiting for our second data collection run, I do want to show off the analysis features. So we haven't disturbed our data collection here, but we're going to go over and see if this will work to show our first run. Oh no, well, we can overlay our first run with our second run. So I am going to show that on the graph at the same time. Note that our axis tools basically let us turn on and off which data we want to display. So our first run, our cellular respiration run, is shown in yellow, and our photosynthesis run is shown in red. And disappointingly, our oxygen here is just not behaving at all. I'm not sure what's going on with that sensor. For right now, I'm going to actually turn off our new data. And I'm going to show you the last run while we're still collecting data on the first, for our second set. That if we wanted to highlight a portion of our graph here, we could then use, notice that this button turned blue. And this is the graph tools button, small icon that looks like a graph. If you click on that, it will bring up a series of options. And if we wanted to find the rate of carbon dioxide production during cellular respiration, we would just simply apply a curve fit. Linear is usually the best fit for these type of data. And then note that you get a little box over here that gives you the equation for the line, including a value for the slope. So that M value is going to be the slope of that line. So you can have students collect that data. And that's in fact what they do in this lab, is they use that graph tool in order to get the slope of the line to determine the rate of either cellular respiration or photosynthesis for each of the gases.
So we have another question.
Yes.
It's a really good one here. We have some issues also with the oxygen levels not changing, which confuses students. What do you suggest?
Oh, that is definitely an issue that we sometimes face. If you notice, actually, it's what's happening right now. Our oxygen is just not responding much. Ironically, I did this earlier this morning with this setup just to make sure everything was operating properly, and had a beautiful run with oxygen data. If you want to avoid the issue or the conversation, you can certainly just do this experiment with the CO2 sensor. The CO2 sensor almost always works wonderfully. Oxygen, most of the time, works really nicely, but it does have moments where it just doesn't cooperate. I think it is worth a conversation with students that sometimes you simply get unexpected results, and it may not be something. A lot of times students have a propensity to blame themselves. They immediately say, oh, it's human error. We must have screwed it up. But that's not always the case. So in this case, it may be that I might have tilted the sensor and the gel shifted, or it might be that the temperature in the chamber is changing a little bit. Obviously, that first run, the chamber was getting cooler because I could feel the air conditioning turn on in the room. So that change in temperature might be causing too much of an effect where our oxygen looks like a flat line, but maybe in reality it would have shifted a little bit if the room weren't changing temperature. It's hard to say what's causing it, but I do think it is a good object lesson for students to know that they don't always get perfect results on every single run.
Yeah, and kind of to note to that of what Sarah said earlier too, is because we have such a discrepancy in the concentration of oxygen gas and carbon dioxide gas in the atmosphere, that also contributes to you just don't see a huge change in oxygen. Like if we're looking at the live data right now, you can see that it was starting to go down, but now it's slowly starting to come back up. So we are getting that production. It's just, again, it's such a small change in actual concentration. It's just not easily visualized, which is why a lot of times it is a lot cleaner and clearer story to often just use the CO2 sensor.
Yeah, that's a great point. Thank you, Colleen. All right, so we're almost to the end of our run here.
So Sarah, how long would you typically plan for this experiment? Would you say a class period? And if students wanted to test different variables, what should they test or could they test?
So they could certainly try, different types of leaves. They could try different light sources. I actually think it's great for students if they have different colors of bulbs or different feeder gels that they could put like cellophanes in front of the light. Typically, you can see a nice difference if you have them try the difference between blue light, red light, and green light, or just plain white light that has all of your wavelengths. That's a great experiment to do to demonstrate. A lot of times students immediately gravitate to the idea that plants are green, they must use green light. And having them actually test those ideas, even those ideas that are incorrect, can be incredibly instructive for them. So that can be a very fun variation on this lab, is to use something like green cellophane, in which case it's nice to use that smaller chamber because the cellophane sheets are a little bit easier to wrap around that small biochamber 250 that I showed you earlier. All right, looks like our data collection is finished. All right, so what I wanted to do now is actually demonstrate some of the features of GA Pro, because it looks like we have roughly 15 minutes left, and I wanted to save enough time for any additional questions. What I'm going to do is show you one of the features. I have GA Pro. It looks just like graphical analysis, regular graphical analysis, the free version. But when you're looking at the free version, you're going to see a little shield with a check mark in it. And if you click on that, that allows you to input a key to upgrade it. And that you would do that, for instance, if you wanted to do the free trial, we would send you the key for that, or you could try it out. You also, if you buy a subscription, it's sold as a yearly subscription. And you would use that key yourself, and you could also distribute that key to your students. So I'm going to show you the first feature, which I think is pretty remarkable, and it's data sharing. And this actually allows you to send data over the web to another user. So I'm going to data share right now with Colleen. So we're going to click on that, what looks like a little radio signal at the top, and we're going to start session. And note that it's going to create for us a little 6-digit code, or 6-character code, ID code. And Colleen, with her version of GA Pro, can now enter that. And she's going to demonstrate that on her end. So Colleen, I'm going to stop sharing.
Yep, I'm going to grab your screen here, and I'm going to share my screen.
So as the teacher.
So I've entered the source code. Can everyone see that?
Oh, yeah, I can see it.
There we go. Okay. I'm going to connect, and it's going to just show me one graph right now. But what I can do is then go ahead and I can change it so I can see 2 graphs. And I just have to change some axes here. And oh, it looks like something's not working. I think I might have found a bug.
Uh-oh.
So it looks like it's showing me oxygen data, but it's not showing me my CO2 data.
Ah. This is a very new feature. I want to say this was released. How recently?
It was released Thursday.
Okay. This is a very new feature.
Yeah. Oh, it's because I didn't have an x-axis.
Ah, there we go. Okay, so it is there.
Okay, and so now I have my own data set here. So what's great is I can now do my own analysis of the data and not basically not have one person do it for me. So I can just look at my one data set of CO2 here, and I can do my own personal data analysis now.
And that's a great thing to point out. So you can demonstrate how to do the analysis for them. but they don't get your analysis. The students do have to do it themselves, which is kind of a nice, that's a nice feature that you can show it on your screen, but when they get the data on their screen, they will need to walk through those steps and actually do the analysis.
Yeah, so I'm going to stop sharing so we can go back to Sarah's screen. You can share your screen there, Sarah, and see how my data analysis didn't carry over to yours.
Yeah. All right. Oh, let's see. It's not showing the right screen. Hang on there. There we go. Or did it not? Did it? For some reason, it's picking up a different screen. All right, let's reposition that.
As I say, I could see the graphical analysis screen just fine.
Oh, okay. On my screen, it looked like it was showing up in an odd place. So.
Yeah, well, now you moved it. Now it's gone.
Oh, now it's gone. Okay. All right. That was very strange. It showed that I was sharing a very different screen. All right. Is it back?
Yep.
Okay. Sometimes Zoom is a little bit problematic. So. All right. So yeah. So the analysis that Colleen did is not showing on my screen because that is only on her device. And when she saves, if she wants to make any notations or anything, you can annotate the data. As I showed you earlier, you can add best fit lines and do analyses like that. You can also simply look at the statistics for a section of data or for the entire graph, and that will give you the range of points within the selected area. It'll give you the number of samples. It'll give you a mean, a standard deviation, and also a minimum and maximum value and the time point at which that occurred. Additionally, you can use that menu to also add things like prediction lines or annotations. So if you want students to add a note to a graph, so we wanted to name this, for instance, they could make that box and then drag that label anywhere they wanted to, or if they wanted to label specifically the line, they could drag it up against the line. I mentioned predictions. A lot of times we ask students in the classroom to make a prediction about data or a particular treatment and what they might see in the data. And they might say something like, it goes up, it goes down. You can ask them to be a little bit more precise and draw a prediction and ask them to actually draw on a graph before they collect any data. So you can say, okay, what do you think this line is going to look like? And they can draw their own line and save it. And they can redraw it if they don't like it. I'm drawing using the trackpad on the laptop that I'm using. But you can have them draw it. They can do it as many times as they want, and then they simply save it. Once they save it, then it appears on the graph with them labeled as a prediction. If they want to get rid of it, they can simply get rid of the prediction the way that they would any run of data. So they can simply toggle it back off, and now the prediction is gone. All right, I wanted to show one more feature of Graphical Analysis Pro, which is the video feature, and I know that we had a question about that. In the Pro subscription, there is an option. I'm just going to launch a new experiment. I'm not going to bother to save what I did. But we're looking at the opening screen here for Graphical Analysis Pro, and note that it says Sample Experiments over here on the right. We have a number of experiments that we've already run and collected. You can certainly create your own. But for instance, if we wanted to do a demonstration of photosynthesis, it's going to take us just a second to load. But now we see the graph appear as well as a video. If we use this little arrow button up at the top corner, that's enable replay. Now your students can actually watch the experiment start to finish. So note that our video now shows somebody in the lab setting up the experiment. And you can create these files yourself. We're not going to have time to demonstrate something like that in this workshop, but we're hoping that we will have a workshop available soon on that. Or you can always call us or email us and ask for training options if you want a longer demonstration of this. Again, you can always email biology@vernier.com. But notice now that the video is synced with the data collection. So we actually see the data start to appear in the graph as the experiment is running over in the video. So this is another option available to you, again, with the upgrade for GA Pro. All right, Colleen, any more questions?
No, it doesn't look like.
All right.
I think maybe we should tell people the cost of GA Pro and how it works, because it's a little different than our previous Logger Pro type licensing.
Ah, yes, excellent point. So GA Pro is a subscription. And as we mentioned, you can get a free trial of it right now. So all you have to do is go to the website and sign up. But a yearly subscription is currently an introductory price of $69. And I believe it continues through June of 2021.
I believe you're correct.
And it's gonna look and behave very similarly to graphical analysis, but it will have those pro features that allow you things like video files and that ability to live share the data out to students on other devices. See, I'm trying to pull up our page right now. So give me just a second. But as I mentioned, if anybody would like to see more of a demonstration of that, you can certainly get in touch with us. Here we go. Okay, so yeah, Vernier Graphical Analysis Pro. So that is an introductory price, $69 for a school or $69 for a college department. So if you're at a high school, the subscription covers the entire campus. If you're at a college, then it would cover your department. All right, well, thank you very much for joining us today. Thank you so much. We really appreciate having you. And again, if you need to contact us, please feel free to reach out.
So have a wonderful day, everybody.